Our director apologizes to all of you, but an unexpected commitment does not allow him to be present at the webinar. So I will have his position to present this series of seminars, the webinar, the Tuesday's webinar, and I will thanks a lot our speaker, today's speaker, Dincer, the doctor and engineer Dincer, for his contribution to this series of seminars. I want only you to remember to everyone that the seminar will be recorded, and that next days you will find the registration, the video of seminars on the YouTube channel of our institution. And so I think that we can start with the talk, and only a few words about our speaker, Dr. Engineer Dincer, is a researcher and is a research group leader at the FIT and IMTEK of the University of Freiburg, and his main interest is about disposable microsystem and the development of bioanalytical materials, sensors, and microsystem, and their contribution with data science and artificial intelligence for various applications, including diagnostic, especially for point-of-care diagnostic and wearables. So these topics fit very well with a lot of our colleagues of our institution and of several institutions of the CNR of Italy. So I thank again Ken for his availability, and Ken, I will leave you the floor for the talk. Thank you very much, and the floor is yours.
Thank you very much, Lucanos, for this kind introduction. So we can start with my talk now. So today I'm going to speak about disposable sensors for next generation diagnostics. And maybe at the beginning I need to clarify what my vision is and how I would like to approach that. And today in today's world, we are living in a population-centered case. What does this mean? So if you get a drug prescribed, which is tested on a small cohort of patients, and actually it's given to everyone at the same dose, independent of the physiological differences of different persons. Besides, also in the sensing, if we develop a sensor, usually we use this sensor for many different approach, many different diseases, and many times we do not care about, and we need to improve that, the specific needs of patients of such a disease, of a different diseases. For example, if you have COPD and would like to develop a mask-based sensor for the patient, so you need to understand what the requirements of these patients are, how they are breathing, and if they have enough air, so that you need to modify your system according to the needs of the patient. So therefore, the future will be a patient-centered care where we need to go from the beginning on developing of a sensor, bring the patient engagement at the very beginning of the development in the design phase at the best, and to understand the needs better and also develop the systems accordingly. It's always much more difficult if you start from top and go down, so it's always easier to go from bottom-up approach. The second point is we are living in a sick care, so the diagnosis and monitoring mostly starts when you are sick and you have a specific disease. However, we have now devices which are called wearable sensors, which we can apply on our body, which can continuously monitor different parameters of our physiology, and so this enables us to have for example, similar approaches which we have able to create in a clinical setting in a normal life, under normal life. So which allows us for a proactive care where the patient or the user can do a lot more and also take care of his or her health situation. So that was actually one of our covers, which we published in 21, which shows nicely how these translation can be. But how we would like to achieve that? So first we work on materials. We are functionalizing different materials, mainly also with tools coming from synthetic biology like CRISPR, the genetic skillers, or penicillin binding proteins, which we are getting from a resistant bacteria to measure antibiotics, for example. Of course, with the increased use of wearables and getting much more data, which we are capable of continuously record the health status. So it's important also to understand what does it means and also maybe compare how was your background, how is it changed, because your background could be also differ from many other people in the population. So this needs also data-driven technologies to help us to understand and find similarities and also disease-related changes in your recordings. And therefore, my group also started to work with data-driven methods. So today I won't talk about it, but I hope that we will publish soon an article where we make our first attempt how we would like to combine sensors with artificial intelligence. The next thing, of course, to close the loop. So it's important not only to get the data, understand the data, but also keep it safely. So in 2 years ago, we also made some talk about it, how we can use ledger-based technologies like blockchain, integrating sensors. What do I mean with that? For example, every person has maybe having on own blockchain, allowing the access of other sites like doctors, and if there is a sensor integrated to this blockchain, so it can report it a change which can be gone to a doctor, and it can even prescribe a medicine which is delivered to you by a smart contract and also paid by the health insurance, by the cryptocurrencies or digital currencies. So this would be, of course, not only the main topic, but we already started to make some thoughts how later we can introduce also sensors into this scheme. And of course, the final goal is always to create biomedical sensors and wearable devices. Here you can see two covers of our sensors, which I'm going to speak about today. Just to summarize what we are going to talk about today is my current research. We do a lot in multiplexing for detecting multiple analytes, but meanwhile we found out it's also interesting to measure multiple samples simultaneously for the same analyte. Then we will look up our crazy project, which is called Love on a Bird, where we are trying to create a wearable device, microfluidic electrochemical sensors to detect hormones on a living animal continuously for 2 weeks, up to 2 weeks. The next thing will be then about our wearable approach, which paper-based breath analysis, where we try to combine the sampling with the simultaneous analysis in a non-invasive way for different analytes like hydrogen peroxide or glucose. And finally, I would like to shortly talk about the OptoAssays, where we use again some tool coming from synthetic biology, the optogenetic switches, which you can bind or release by changing the wavelength, and combining with protein of interest, you can create dynamic assays, which you can control with smartphone. So this will be the program of today. Starting with the microfluidic multiplex biosensor. So this is our let's say not the main business, but the major research area. So here we target different antibiotics, inflammation markers, nucleic acids, and hormones. Here you can see some of the multiplex designs which we made in the previous. Later, I will also show you the one that we are currently using. The first area of application where we apply this multiplex sensor is therapeutic drug monitoring. So here, after the sample collection, the sample analysis currently takes around a day, which based on chromatography or immunoassays, which are mainly requiring sample preparation or long turnaround times. So which means actually, if you have a day after the sample collection, the doctor can adjust your dose immediately after a day, which means for many diseases, unfortunately, too late. So we need apparently much more faster concentration adaptation in case of an over or under dose. So the idea is to break this loop and create an immediate detection. So therefore, our first approach was a personalized antibiotherapy, where we do management of the therapy by measuring the drug concentration, assessment of the therapy success by measuring the inflammation markers, and finally, we would like to combine that also with the prognosis of the organ function. So it's a whole program of a therapy. So currently, we are still in the first phase, management of the therapy. So I would like to show you the latest publication, which we made by the end of 21 and beginning of 22, where we made a pig study where we use overdose and normal dose pigs, received samples from exhaled breaths, non-invasive and invasively. So we have plasma and whole bloods, and invasive, and also saliva, urine, and invasive. So here you can see our electrochemical approach chip, where we, independent of many groups working on the electrochemistry, we do not immobilize our assay on top of our working electrode. So we have a specific area in our microfluidic channel, which we call the immobilization area. And so these two electrochemical cells and where the electrodes are and the immobilization area are strictly separated from each other by a stopping barrier, which is a Teflon coated barrier. So the idea is we introduce the sample through the inlet, which runs through the capillary forces automatically to the stopping barrier, and the incubation takes place. After the incubation, we suck it from the same inlet, the same solution. So the electrodes do not have any contact with the samples, for example, which allows us therefore to work all kind of samples. So here maybe I need to note, of course, the whole blood, it was heparinized. So but we can up to date work all kind of sample types with our chip. So everything is possible. Looking up to the assay that we use, I already told we use a penicillin binding protein which recognizes the beta-lactam ring of beta-lactam antibiotics, which is actually usually not a good thing if you have a bioreceptor which is unspecific for all beta-lactams. However, in the case of therapeutic drug monitoring, we always know what kind of antibiotic we gave to the patient. So therefore, it's in this case even better because we have only one assay to detecting all type of beta-lactams, which is a little bit problematic if you would like to do it with antibodies. So there you need to very specific antibodies for each type of beta-lactam. So this study actually won also a prize, which I would like to highlight. Actually, this would be also the only prize that I would like to highlight because this is won by my PhD student before finalizing her PhD. So therefore, it's also very valuable for me. So looking up the highlights of this study, after measuring all kind of the measurement, we showed that whole blood measurement is possible. So that was the idea. Is it possible to conduct a glucose-like measurement, like a finger prick glucose measurement from the whole blood? So it looks like that in all three cases, we see clearance behavior. I have to point out, so we have a competitive assay. So after administering the drug, the signal is decreasing. And after 4 hours, the antibiotic is cleared from the body. So which means that this concentration reaches to 0 to the basic initial status. And here, maybe one point in the normal dose, we see an difference to this curve. This is the reason actually we found out because we did it while the animal was in operation, so it gets propofol as an anesthetic. And we found out in this case that the patient, the pig, was about awakening, so they gave immediately a dose of propofol, and while exactly at the same time by getting the antibiotic sample. So we checked actually if you wait 5 minutes, so everything will be fine. So that, however, it's also good to know under which conditions you need to measure the samples. So if you would like to have a propofol injection, then you better need to wait for 5 to 10 minutes to be on the safe side. But the main highlight of the study was, of course, the measurement in the exhaled breath condensate. So up to date, it was possible. Some people show that they can see some residues. However, we could nicely show that we have also can see a clearance behavior for all 3 samples for the EBC, which was really for the first time in the literature to measuring piperacillin tazobactam in exhaled breath and seeing also immediate correlation with the blood sample. So, which we believe, of course, through the good blood circulation in the lungs, so that the lung fluid gets very fast the concentration of the blood levels. So we also validated this with the plasma, which is the gold standard for the characterization. A big point, of course, in the sensing is we always need to benchmark your sensors. So you cannot say, I measured it and that's it. So you need to go and compare it with the gold standard. Here we did it with HPLC. The main point is the exponential decays are very similar, which shows that our system is very well correlates with the gold standard. We also do an internal calibration then, because that was the single channel version, so with the multi-channel version showing that the benchmarking also works for these sensor type. I would like to, so currently we are working on different antibiotics, different beta-lactams with the same system, and also would like having the idea as a breath diagnostic. An important point will be how reliable is it? And we need to understand what are the limitations. For example, if you have a history with your lung, so some diseases, does the correlation still there? So this is something which we are currently looking for with our system, and also different antibiotics. So I hope that this will be also published this year. But I would like to close this part with our latest article in Lancet Infectious Diseases, where we propose a model, a 3-level model, together with medicine. So therefore, I'm very happy as an engineer, as a corresponding author, as an engineer, having working in a multidisciplinary environment to creating this model. And the idea is usually you got the antibiotics via bloodstream, which we call upstream. So then it's actually distributed to many different areas. And for example, if you have a lung, so you will get it into lung for fighting the tissue, the bacteria in the tissue. But so the idea was actually we only measure the concentration. However, we mainly interested actually the concentration in the tissue where we are really needed. So the idea here is it possible to make, for example, an upstream measurement and also a downstream measurement, which means actually from the biofluid, like in the lung fluid, in the case of a lung infection, possible what's going in and to understand what's going out in the lung fluid, to see the flux, and also to having an idea about the real concentration in the in the tissue, in the lung tissue, for example. And this could be, of course, also used for the different areas of where you need to find the infections, bacterial infections, like in the skin or other areas. So and this combines, of course, also the wearables with our point of care approach, where you can measure, for example, the blood concentration, and also with a wearable looking up for the downstream concentration. So the second area, and this is actually the area I got quite famous with, is the CRISPR-powered diagnostic. So we started with that in end of 2017 and worked a lot in 2018, which we published later in 2019, our first electrochemical CRISPR biosensor. But before explaining it, I would like to give you a short overview of this approach. So once you have isolated and purified your DNA or RNA, you could use two different modes. So one is called binding-based CRISPR diagnostic, where we have a Cas enzyme which is deactivated, which means it cannot cleave, it can only recognize the target. And my colleague Kian Aran, meanwhile in the UC San Diego, created the CRISPR chip where they bind it on a surface of a graphene field effect transistor, and they look for genetic mutations in the whole genome. And it was possible to detect it without any isothermal amplification. So once it recognizes the mutation, it binds to the Cas, and of course the whole genome has a huge charges bind onto the surface of a field effect transistor, giving you a measurement signal. However, this is very nice approach for longer targets. However, if you are working with microRNAs, which are specifically around 18 to 20 bases, then the charge that you bring on the surface is not that good. So you need to find another approach, which is the cleavage-based CRISPR diagnostic, using the whole capability of the Cas enzymes. So, and here maybe skipping the part with the amplification, if we have the target, so which is recognized by the CRISPR-Cas complex, then you can, for example, use a fluorescence molecule with a quencher and as a reporter, and once the target is recognized, it cleaves all surrounding RNAs, which means this is quenched, and then you can get a fluorescent signal. So, and this is of course good because you have a time-dependent reaction. You can wait longer to get better sensitivity. And our approach will be also basing to the cleavage-based one. So, but before going into that, within the last couple of years, of course, CRISPR get very hyped. We saw many different nice examples of CRISPR diagnostics now. To be honest, we were at the beginning a little bit lucky because you can't not buy at that time Cas13, but we are working very closely with the synthetic biology department, so we could able to produce our same enzyme. So today's actually you can find different types of Cas enzyme, of course the Cas9, which is used also for the genetic purposes. For the diagnostics, however, we use Cas12 for DNA, double-stranded or single-stranded, Cas13 and for the single-stranded, and also Cas14, again, which is a little bit smaller than the Cas12, sorry. And however, there is also a Cas-P, which is found by the Nobel laureate Jennifer Doudna's group. So this is interesting because Cas is usually a bacterial defense system. However, this Cas is coming from a bacteriophage. It's coming from a virus, so it's never used for the diagnostic application. I don't know, maybe there is a patent issue, but if you want to start and there is no patent issue, so this would be, I think, an interesting thing to test it. So but now I can also tell what we did in 2019. We use it for microRNA diagnostic on an electrochemical microfluidic chip. And maybe just to explain you how simple it is to use it. So you need three components. You need the enzyme that you are looking for, and in our case, it's Cas13a. You have a CRISPR-RNA, which is actually kind of the key lock, so that the blue part is complementary to your target. And once it's, of course, these two comes together with the target, it starts to cleave. And we use also a reporter for our reaction, which we later also labeled with an electrochemical marker. So we use also fluorescein with biotin. And the only thing that you need to do is put them together with your sample into an epi, wait for a specific time at a specific temperature, which depends on the Cas enzyme that you use. If there is no target, there is no cleavage, you can build up an assay like we did with an enzyme-coupled antibody against fluorescein into our chip. And in that case, of course, we've got a high signal. So if you have a target, it starts and cleaves everything in its surroundings. So in this case, the biotinated site still can bind. However, the signal generating part is washed away. And in this case, of course, we got lower signal. So it behaves like a competitive assay. However, I have to say it's not a competitive assay. So we don't have a binding places. So it's just a cleavage-based device. So it's just the signal is disappearing. And this is just actually an assay design issue. So you can also reverse that if you want. Now you see a lot of components in the system. And the only thing that you need to change to target another RNA is the blue part of this CRRNA, which is an input to our system, which means actually you can have one assay for measuring all kind of microRNAs or other RNAs. So this is actually the biggest power of CRISPR, and it's very simple, so you can even do it. Okay, let's taking out the RNase contamination in the case of Cas13, but you can even do it at home, actually. You don't need much. So in the first work, actually, we concentrated us on microRNA detection for brain tumors. We showed that in the clinical samples that we can measure it in children serum microRNAs, which are without any target amplification. And there we had a sensitivity around picomolar range, lower picomolar range, within 4 hours. Today, of course, to spare also some time, I would like to show you the latest publication, which we did actually around a year ago, for COVID-19, where we also where we actually first reduce the time from 4 hours to 30 minutes and also improve the limit of detection from picomolar to the femtomolar, again, without using any isothermal amplification. And how we did it, I will explain shortly. But in this work, we did a little bit more about that. So we combine also our antibiotic assay into the same platform with the COVID-19. First of all, that was a strategical move to show that our chip is capable of measuring different assay types, for example, nucleic acid together with protein-based assays. Of course, it would be better if you would like to measure antivirals. However, for COVID-19 patients, actually worldwide 75% also received antibiotics in order to prevent any co-bacterial infection. So we also worked a little bit around our point of care measurement approach, where we tested an NFC antenna and NFC potentiostat actually with a micropump to measure our chips with a smartphone or other smart devices, which we can also power up and do the measurement. Here, just to remind you, both assays ends up with the same enzyme, which means we convert glucose into hydrogen peroxide. Due to the spatial separation of each channel and the detection cell after each measurement part, there is no issue of cross-contamination, so you can really work nicely on that. And the idea was once you get tested positive, the drug concentration will be measured, and we would like to keep your antibiotic concentration, for example, above a limit which you can required for the fighting the bacteria. And once the, of course, therapy ends and you are negative, so you can leave the hospital in that case. So about the assay optimization, how we get 1,000 times better. So it was quite easy, surprisingly. So we were using an Cas ortolog, which is coming a specific bacteria. We changed this to an LVU Cas 13A with the same reporter first, and we already saw the first thing that our nanomolar signals are going down. And then we read, of course, the LBU Cas 13A likes to cleave uracils more. So we made actually a reporter which is basically consisting of 20 uracils. And with this, it was possible to measure also less in the femtomolar levels. So then we calibrated our system for E gene and the IP gene of the SARS-CoV-2. Both delivered us in a lower femtomolar limit of detections, which are statistically calculated. For those who likes copies, then femtomolars, so it's around, we are speaking around 2,000 to 8,000 copies per microliter without any isothermal amplification. The good thing is actually on a longer genome is you can also target say two areas simultaneously. So we did this as well. So it really got an amazing limit of detection. However, we didn't test it in real samples. So therefore it's and also our main focus will be staying also as a microRNA, so which are short. So therefore it's with precaution. I think it should be handled with precaution. However, what I can say, we show that multiplexed and amplification-free detection in femtomolar range within 30 minutes. If you combine them, it might be possible also to measure where in even 1 copies per microliter. So we tested also in real samples with our approach. However, we, as I said, separately. So we have a positive control, which was a microRNA, which we artificially gave to the sample. We had SARS-CoV-1's at the IP gene as a negative control to see of our height, signal height, and also at the IP and EG signals. So, and what we see in the patient 1 and 2, actually, that we can see a signal decrease. However, again, I need to say CTs around 18 are actually patients with high viral load, which you can also detect with lateral flow assays. Here, I need to point out the next issue that we are currently working on is the CRISPR is, of course, the targets, the calibrations are done in PBS with the target only and the reporter. So here, however, in the real samples, you have lots of different RNAs which are also cleaved. So Cas does not say, okay, I would like to cleave your reporter, so it cleaves everything. So currently, we are working on an approach how we can introduce the sample, activate the Cas, and give later the reporter to cleave for the cleavage. So this would be our challenge to get the same sensitivities also in the real samples. Also, we did this part with the antibiotics, as I said. To spare some time, I will jump in. So it really shows that once you administer the antibiotic, the signal drops out, and after 4 hours it comes back, which theoretically shows nicely that we are able to combine different assays onto the same platform. And here you can see also how our chip is built. So it's a polyimide-based platinum patterned chip with SO8 coated, and we create the channels by dry film photoresist, using dry film photoresist for our approach. So if you are interested in CRISPR, and don't know where to start, this is just an outlook for you. So I really believe that you need to do amplification-free detection. If you combine it with isothermal amplification, then most probably I would suggest to go with the isothermal amplification, because it doesn't have its advantage anymore. Having easily scalable, then you will need much more actually than a normal one. You will get a little bit more specificity, but I don't know if it's really its value then. Another point is multiplexing. So we showed already how you can do a drug together with a virus, for example. However, what you could do is also with antibodies or others. And also what you could do, for example, I like this idea. So this is actually one of the publications that I could find in the literature. So what you could do, of course, that using an aptamer, recognizing a protein, and this aptamer could trigger or stop triggering a CRISPR reaction, where you can use CRISPR's amplification capability to make protein assays much more sensitive, for example. And the next thing is, of course, the wearables. So there are lots of space there for CRISPR. Actually, the group of Jim Collins at the MIT already show a face mask with CRISPR for COVID-19 detection. However, this is the first and actually one of the only examples. There is a microneedle with Cas9, but so there are really only 2 examples. So, and as you can see, there are lots of platforms where you can apply that. So there will be an interesting survey, I think, if CRISPR starts really going into the wearables. Finalizing this part with the microfluidic chip, we have also an approach, as I said, it's a sidekit crazy approach. We started with small chicken, small birds. Now we are on the chicken. It was not that easy to measure hormones. So this is a code which you can also buy in Amazon, where the system is currently integrated. The idea is to get 10 microliter blood every 2 hours and do an actually very normal competitive immunoassay for testosterone detection. Here you can see one of the some of the previous versions of our system. So here we have multiple microperistaltic pumps to control the fluid, because it will be a line on the wing continuously for 2 weeks, and then the blood will be diluted, then mixed, etc., etc. So we need a couple of these microfluidic pumps. We also change our system a little bit. We separated the electrochemical cell from the microfluidic chip. So this part is like now the old film rolls, if you can remember. So once you measure it, the next one will come up. So this is the idea behind it. And with such an approach is possible then every 2 hours for 2 weeks to measure it. Here you can see one of the animals that we installed a system with after an operation. So you need to still do it, of course, while the animal is under anesthetic. So here you can see also after the operation, so how it behaves. So that was also an issue for us to understand, is it possible that the other chickens, pigs, and the other chickens, because there are lots of tubing, so if they are getting really attacked from each other, play with it, how they feel. So that's actually currently the stage of this project. It took a little bit longer than expected. We have some assay problems, we have some as blood problems. However, it's not because of the coagulation, surprisingly, but so I hope that we will soon also have a publication. However, there I cannot give a specific date, unfortunately. The second approach that my group works on, which is also coming from the time that I was at Imperial College, is a variable breath sensors, where we are trying to create a simple and low-cost approach for simultaneous sampling and analysis. So here we use paper. However, you can use all kinds of porous materials. So the idea was it's a substrate for electrodes, so you can bring the electrodes on. You can use it as a solid phase. You can immobilize biomolecules, or you can absorb enzymes, for example. You need to put salts, of course, to make electrochemical detection, but once you exhale, it gets wet and you can do the analysis. So we call it also as a solid electrolyte as well. And of course, breathing as a reflex, so you need to breathe 15 times a minute. And therefore, it's a non-invasive sampling method. And the idea was actually to get rid of condensation, so just to do an direct detection on the paper after the collection. So that was actually before the pandemic. So I have to say, because after the pandemic, it was kind of much more easier, I think, to start with, because once we created the device, we really thought about, okay, how we can really manage it to people having a mask. So we started also with an extension of a mask. So we did the first work was on hydrogen peroxide detection as an inflammation marker. The idea becomes, of course, we have a differential setup. So we have a sensing electrode, which is sensitive to the analyte, together with all other noise. And we have a Planck electrode. This is for all noise. And because you have, of course, a very challenging environment, you have mechanical movement. Then you have, of course, a temperature change because you exhale 35 degrees with 100% humidity, but inhale the room temperature and also the humidity. So, and also in the first approach, we choose, I would say, also a very challenging target, because hydrogen peroxide is very low level in exhaled breath. So therefore, we could measure it in a simulation, in a simulated setup, so where we have a pump where it's really condensates the hydrogen peroxide and brings to the sensor. Here you can see, for example, one of the measurements we did. So it can measure continuously here 2 hours, for example. Now we are trying to extend it for glucose detection. So this is kind of giving you an idea. So you only need to make wax printing and for creating the isolation, and you could just separate these two areas with the same electrode, in this case with the mediated carbon, Prussian blue mediated carbon. And on one side you can put the enzyme, on the other side no enzyme. So on one side you have the glucose signal plus everything else, and on the other side is just for the reduction of the signal. So here actually we have also some initial results. However, this is not that, so we are still working on that. Just to give you an outlook here as well. So face mask integrated sensors, of course, after the pandemic, it became also an issue. You can do metabolite measurement with enzymes. You can do chemical measurements directly with the electrodes. You can do pathogen measurements, for example, even integrating the sample extraction. You can even combine with CRISPR. And here you can see actually one of the images I made it 2020. Once Nature Biotechnology contacted me for our CRISPR work for COVID-19. I told them that we have also a breadth approach. So here you can see it's a really working prototype actually. So therefore with cables. So, and after 4 years, I would also tell you I would do now the system a little bit different. So more rigid mask like FFP3 mask with wireless communication with the smartphone still. But however, this was actually a real one, so which really worked in total. So, and of course, during that time, we also made some considerations about, as from the engineering side, how we can create new devices for wearable breath analysis. So masks are now kind of given available in the literature, at least in the academia. What will be the next platforms? And there we propose actually some ideas. So one of them is an in or under nose patches, which can catch the exhaled breath and do the condensation and also do the measurements. For example, we have really good patches for sweat analysis. You can reverse engineer it for such an approach, for example. Then a little bit more, much more academic would be the intrahepatic patches, where you can even use maybe metabolites for self-hovering. And actually the one that I like mostly, and it was unfortunately patented already. Someone put sensors into a vaping device patent, unfortunately. Vaping devices. Why that? Because these can enable us to maybe inhale drugs. And what about to integrate also sensors, for example, for a lung disease, let's say asthma, you get a drug, and for a specific dose, after an hour, you can exhale, do the measurement, check if your drug concentration in the lung fluid is still okay. And if not, even though maybe it can be connected via internet so that the doctor can tell you or automatically change the concentration of your drug. So therefore, I really interested in. There are, of course, some challenges with saliva, and so we put actually all of our ideas on that paper in Nature Reviews Bioengineering last year. In the final part, I just want to lose some words about the latest approach with the OptoAssays, where we use optogenetic switches, so one phytochrome B and phytochrome interacting factor. And as I said before, you need only change the wavelength, and then they are either in an inactive or active stage. Our idea was to create a wash and pump-free bioassay because you have the biodirectional movement. How should this function? So we have a sender area and then the receiver area. Both are nitrocellulose currently. So you can immobilize your optogenetic switch. And here, for a proof of principle, we made an HISTEC assay with an anti-his antibody with labeled GFP. So the idea was, once you change the wavelength, and put the analyte in a protein with a histang, these two are competing for the binding spaces, and once you change the wavelength, so it binds back. However, of course, I'm aware of the word diffusion, and so we are currently also trying to accelerate these back movement also with other technologies. So here you can see the real measurements results with analytes, without analyte, and also with binding. So we created for these and black box, actually a photo box with a smartphone with Arduino, where we have the far red and red lights, and also of course our camera with a filter for the GFP, and we did also the calibration. So this paper is currently in review. Hopefully we can also publish this very soon. So we got a very good limit of detection for the case of the proof of demonstration. However, you also see that we have actually quite huge standard deviations. So this would be now actually after showing the concept. So this would be now the advancing the system and also bringing into real assays that are much more of interest. So with this, I'm end of my talk. This is my group in Freiburg. So this is our faculty. I don't know if you have been in the technical faculty in Freiburg. I need to say 2 more words so that I can show you the Münster of the city, where the city center is. Of course, I'm the frontman, so there are lots of people who contributed to this research and to this nice results. My students, Jaron, Midori, Nadine, Regina, and also thanks a lot to all collaborations, also collaborators. So I believe science is about sharing information. Therefore, you can find me or my group in the following social media channels. And if you ask yourself what social media got to do with science, I have a talk on YouTube. Maybe you would like to, maybe I can convince you about it. I think with this, I'm end of my talk. Thank you very much for your attention.
Thank you very much. Thank Dan for canceling for a very, very nice and impressive talk. And so I will leave the floor for people who want to make some question to Ken. And if you want, you can raise up your hands. Okay. I see a question from Punk J. Okay, you can unmute if you want, and you can have. Sorry, I unmute you probably. Okay. Can you unmute Punk J?
Yeah.
Okay.
Can you hear me?
Okay. Yeah.
Yes.
Very nice presentation, sir. Very impressive, really very impressive presentation. Sir, I want to know one thing. I'm also working in same direction, like in biosensor and everything. So, sir, you have developed several wearable biosensors. So, means generally when we are performing the biosensing system, then we are adding some electrolyte. So in wearable sensor, there is some need to add electrolyte, or there is no need to add electrolyte. How this is detecting the things?
You need to give actually some salt to the paper, so you need to really treat it. However, other than that, you don't need to give actually. Okay, you need in the lately phase we also give a buffer. So we add actually a buffer, which is getting then because we use actually Prussian blue, and Prussian blue is much more stable if you put potassium, I think. So therefore we have a and actually potassium, yeah, and so we add also a buffer to the system as well. So, but what you do is basically actually after the production, you add the buffer to the chip and let it dry.
Yes.
And usually in the case of, for example, glucose oxidase, we give glucose oxidase in the same electrolyte so that we can just immobilize it and let it dry. Dry means, of course, on the paper, the humidity. But anyway, so that would be the small treatment, yes.
Yeah, one more question, sir. Sir,
Sure.
you have developed one chip on board system. So for that, sir, where will the display? It was connected with Bluetooth, or display will be on that wearable sensor only?
No, it
Where I
will
can
be
see our signal.
there will be no display in that case. So the area is at the beginning having some remote readout. So I have to say we really started with very small birds. However, there is some problems with the blood getting out the blood. So that's kind of difficult because the blood pressure of these birds are not that high. And even though if you apply with sucking with the micro pump, so it's not that easy to get it out. After the operation, always it works great because they are stressed. So, but our goal is not to stress them. So that's a little bit
So you have mentioned that after 2 hour, every 2 hour we will collect the blood, 10 microliter around blood, and then we will measure. So after 2 hour we have to do blood collection manually, or automatically it will do?
It will do automatically. So it will have it will have some as I said, these motors in its own control. So it's doing it automatically. There is also a dilution. Of course, we are not measuring directly in whole blood, so it will be if you buffer it first and then done the measurement.
Thank you, sir. Thank you so much, sir.
You're welcome. You're welcome.
Okay, so thank you very much, Khan. And if have you a little bit time for other question?
Sure.
Okay, thank you very much. So I invited the audience to have a question, curiosity, and so on. I have someone, but if okay, otherwise when we are waiting from people, I have raise up my hand so in order to show. So one question is this one. Now we are a lot of sensors, or say we have a lot of proof of concept of sensors for collecting information about our health from several point of view, from several biological fluids and so on. And this is increasing very in the last few years, and this is a lot of information and a lot of site or platform where we keep information. And what do you think about the future, the trend? Because there are trends that solutions that are oriented on wearable, other solution on point of care. Wearable have several problems, like for example, as you know, the question that you have to be a lot as possible as more impacted with the people, so the biocompatibility. So there are most of challenges, but when you are something in your body or close to your body, probably you can have more information, more rich information about the analytes or what you want to monitor. But why point of care is easier, but probably not good for some kind of pathology or so on. But you have to fight, for example, with the market, because at the moment we have several proof of concept, but we have to move to some. We are also engineers, so we have to move also to some kind of applicable, so some device that could work, that could be able to move to a volume production. What do you think about the standard trends, and what could be the route that are more close to the commercial version of this several approach that you can see on the literature? What is your ideas?
From the point, the comparison between the point of care and wearables.
Yeah.
So actually, I'm not on one side. So it's actually, I believe it depends on I always use this term, but it depends on the application. So we also discussed it also with my PhD student today. So if you would like to measure antibiotic concentration, you don't need to be always on a body. So sometimes it's better to get the classical drug measurement on blood with a device, and because your physiology is currently changing. For example, you made a sweat patch for drug measurement, but you have sepsis and you gained your physiology changed drastically in a day, and then you cannot expect to this patch would work as good as before. So therefore, I think there's a good there is actually, you need to balance it very well. So I think we need both. It's kind of like, it's always asked, I was always asked, would the lateral flow assays disappear when your sensor would come out? No, because they are really cheap, really simple, and really good. And before you find similar things, so it's not possible. But what we saw also through the pandemic, we need not only one system, we need everything we got actually. So therefore, I believe we need both, but it should be discussed on the application basis, and whether the patient status or the disease requirements, etc., etc. To your other question with the variables, it's a little bit more difficult. So I really like the quote of Anthony Turner, who is the who made the Biosensors by Electronics journal. He said once, actually, we are measuring not we would like to, but what we can. So that's actually still kind of the situation. So we are trying to break this circle. However, we really measure what we can and not what we would like to. So there are some works by the group of, for example, Weigao from Galtech, showing some other analytes than electrolytes, but it's still at the very beginning. So I think there you need to really find an application. You see also in our platform. So we made the platform with the bread, and we are also looking for applications which is suitable. So we tested H2O2 because it was much easier, to be honest, to test our system. Now we thought, okay, glucose could be interesting, and looking for that, and maybe we will change again because we will find other bugs or problems with the application. So it's similarly also on the sweat. It's also, up to my knowledge, there is only one disease where you can use sweat for the diagnostics, and you do that also very classical methods and not with a wearable sensor. So I think it's still, but I think it will be coming also now a little bit faster. I believe in the wearable side, one of the biggest challenges is we don't have biorecognition elements, which allows us to measure continuously. Because the big thing on wearable is you can measure continuously without disturbing your user. And there, I think there are still some approaches, but I think we need, except the enzymes, we don't have any solutions. So enzymes is nice because they just catalyze and work further, but
thank you very much. Thank
You're
you,
welcome.
Ruska. So I saw a question from Samson. If you want, you can make your question. Thank you very much.
Okay, did you hear me?
Yes.
Yep.
Yeah, okay. Thank you for your presentation and being ranked for the top scientists in the world. Already have said in LinkedIn profiles. And I would like to ask some questions regarding to CRISPR-powered diagnostics, which is an offering advancement in detecting viruses like diseases and pathogens. So my question is going to be on the sensitivity and the specificity. How specific is that in able to distinguish? To distinguish between the closely related sequences and remain as a significant challenge? And is that can be possible to get off targets? And how did you handle that?
It is possible, of course, to get off targets as well, unfortunately. However, I have to say the system is highly specific. However, so we tested it also with single nucleic base differences. For example, we measured microRNA 19B, and then we tested it against also microRNA 19A, which differs only one base in the middle. And we saw for very high concentrations in the nanomolar range that also microRNA 19A triggers the CRISPR reaction. However, the good thing is actually, if you know your problem sequence, you can really add purposely some mismatches to increase or additions to increase your specificity. But of course, if you don't know the problem molecules, so that would be a little bit problematic. In our case, we thought in our case it wasn't that bad because in microRNAs you look for families, so for different microRNA families, so they are about 6 to 9 And in our case, these two, the A and B, was the same from the same family, so they both was upregulated. Therefore, it wasn't that big problem. However, you need to definitely search for it. I can say, and also many reports show that it's better than many isothermal amplification techniques. So therefore, people are trying to combine those. But as I said, I'm on the amplification-free side and stay there. So, which means actually we use a lot of amplification, but not target amplification. So that's maybe the point.
Okay, thank you. And just follow-up question is also your lab is more focused also functional materials. Did you use a specific nanoparticles that can help you for sensitivity or specificity on especially sensitivity? What kind of nanoparticles, or did you like call nanoparticles, since it depends upon the size and the sensibility also it can give us more sensitivity?
Our electrodes are out of platinum, so which is actually quite catalytic active. So therefore, we never use further metallic nanoparticles. Lately, we tried some MIPS, actually molecularly imprinted polymers, in order to replace the antibodies, because antibody is the most expensive component of our assay, and we don't need it usually if we can combine the enzyme with another technology. However, with this, it was an hydrogel-based MIP, and it didn't work well, so it was very much unspecific adsorption, which made us a lot of problems because you don't want to have signals if there is no reporter there. So maybe in future we will further try other nanoparticles, but up to date we just tried actually what we can get about the assay and the system itself.
Yeah, vielen Dank. Danke.
Thank you very much. And I want only a last question, waiting for eventually some other question. What do you think about the fact that, as we talked before, if the idea is to have much more information about our health in continuously or with high frequency during time. So we need several devices. We need to power this device. We need to have communication wireless, usually to have this information. So we need a lot of device on our body or close to our body. What do you think about the possibility in the future in order to reduce this budget of power and a lot of things that we have to draw to have on our body, to have a kind of combination between the inorganic and organic chemistry? What I mean, a kind of interaction between the electronic part of the device and some kind of organic part that could be, for example, I remember some example in the past where someone tried to grow a neuronal cell on some kind of platform, PCB, and so on, in order to have an interaction between neuronal cell and the biological chemistry with the inorganic chemistry. What do you think is possible to have some kind of integration in order to reduce this, in order to move the electronic part to the organic part and reduce this budget required for having this information and are close to the net? I'm talking about fuel because there are a lot of information that we need and possibility to it. But if you think also in our body, we have several electric signals that are moving from several parts of the body. And electric signals are also between interaction between cell of several type of so if we are able to interact with this, probably we can in the future have a more interaction, so reduce this need of devices and to have same information but in a more less invasive way. What do you think about this idea?
Maybe
it's a future
maybe
idea, yes, but
from the short term, I really believe
it's very difficult, yes.
For example, that we can combine variables with multiple sensors, like, so therefore I also try to use existing gadgets like smartwatches, which you can integrate a lot more, like you have, I think already blood pressure, temperature, so you can do really you can integrate much more, and in total I believe
the same,
as you said, there will be a network of variables. However, I don't know if they will be always on our body. So maybe it will be some on the shoes. If you wear them, you will get connected. So it will be interestingly. Regarding and powering up, I currently think about like NFC, where you use other devices to power things up. However, what you said, to be honest, I never thought about it. Is also very interesting. I don't know how we can use the energy of our body. Okay, you can do like biofuel cells with glucose, octane, so that would be possible. Maybe this would be one way how to connect that. Or maybe, of course, some implantable devices. So that could be also, I think, an interesting approach. However, I'm always on the side of going step by step. So maybe first variable.
No, it's only a trend. If you think that would be a possible trend or a route that you don't have to check to try.
You can do also much more easier sensing. For example, I also read some articles about where you drink a solution, and once it just goes out of your body, you got a signal. And this is kind of zero electronic, but actually you see what kind of disease you have. However, I have to also say, if you use currently the word nano, everyone is afraid of, kind of, in the community. So I think this should be also considered kind of, because people are really thinking that we are implanting chips on them. So that's kind of, I'm trying to convince them that if this is possible, I would be homeless and also jobless. But anyway, so so this is, I think this would be an interesting topic. And I know that this is done on cell basis, like nerve cells or brain cells. But I think I would go in the other direction, how to implement them onto our body. So, however, as I said, I never made lots of thoughts on that. So, therefore,
thank you.
You're welcome.
Thank you very much. So it's quite close to 7 p.m. So we have much more of your time, and so it's very pleasure for us to have your contribution for your talk and your question and answer after that. So I will thank the speaker, Dr. Engineer Dincer, and I will wait you for other next event of the series, and have a good afternoon, and see you soon. Thank you very much again to everyone.
Thank you very much, Maganas. Bye-bye.
Thank you. Bye to everyone.